right, fighters, welcome to Reptile Fight Club. We're here for another fun little episode. We're going to be drilling an expert, Samuel Kerwin, but he is not here yet, so we're going to kick off the show, and he'll get here soon. So figured we'd catch up a little bit. So how you doing, Rob?
I'm doing great.
Excellent. You have a good trip out southeast. You're going to record on NPR, I assume?
I don't know. I don't think we've made a plan as to it.
Oh, okay.
But yeah, we went down to basically in and out of New Orleans, checked out some longleaf pine, loose sand upland habitat in central Louisiana.
Nice.
Checked out the area where Louisiana, the sort of the L corner of Louisiana and Mississippi, and then went over into some longleaf pine upland habitat in Mississippi, and met up with Zachary Gray, who we had on the podcast a couple months ago. That was really good.
That worked out, yeah.
Yeah, so that was great. Principal goal of the trip, I suppose, was a canebrake rattlesnake. And Brandon, so went with Eric and Owen, as you alluded to, and Brandon Valentine, our buddy from down in Phoenix Tucson
area.
Yeah. And Aspen wanted to come, but had travel difficulties and was unable to see it through.
Yeah, so he messaged me. He said he was bummed out about that. He was on like standby or something with family. Yeah, that's a rough deal. Yeah.
So yeah,
he gave it the old college try though. I think he said he waited a couple flights and then he's just like it's probably not going to happen, so he gave up. But yeah, the
good news was good news, bad news is that he was still at home, right? He was at the airport at home. He hadn't made it halfway and then got stuck, which is good.
Oh yeah.
But it was certainly an adventure. So Eric and I had gone, I suppose, skipping to the main point of the thing was, canebrake rattlesnake.
Yeah.
On the second day, we were in that corner, the L corner at Louisiana, Mississippi. And, Eric and I had gone one direction within this, national state park. Yeah. And Brandon and Owen had gone another, and we'd both walked for an hour and a half, something. It could not be further from one another. And then we started wandering back, and then finally the calls came through from Brandon and Owen that they said, we found a timber, or canebrake, although I would say timber rather than canebrake, and it really did get into from my perspective, I think the northern stuff and inland stuff is timber. And it's really a function of coastal form is what gives you that canebrake presentation. So they did see a really large gravid female who then dipped into a tree hollow and stopped. But I was not particularly optimistic. So we had about 45 minutes to walk to him. And I was not running because I was like, well, they said it dipped into a tree. Not particularly optimistic on it. And that was totally accurate and correct. It was well and truly underground, so we had fully missed it. So yeah, that was always, right? That's kind of an interesting situation.
But they got photos and everything, so you could.
They got a 10-second video.
Yeah. Oh, okay.
In which it's very clear that it is sort of timber form, right? Not all that dissimilar to what we've seen in the Northeast.
Yeah.
So yeah, it was a bummer, but it could have been worse. So we. Basically, we'd spent the first day in central Louisiana. The big learning out of that habitat was, the chiggers there produce, at least in me, an atomic reaction, so that both my legs just got these sort of instantaneous large welts as opposed to small dots.
Right.
Yikes.
Yeah,
so really, really something.
Cajun chiggers. They'll get you every time.
Exactly right. So now I guess we are coming up on or are at 10 days out from having been there and gotten those. So they're just now sort of on the mend, I would say. We also, Brandon, being a desert guy, I don't know that he's had much, if any, tick exposure. So the first contest of the week was the count of ticks that we each had.
Mm-hmm.
So we were counting throughout the days how many we were pulling off each night. You'll be glad to know that I won the challenge.
in suitable form,
Yeah,
typical
right.
form, I guess. Yeah.
But at a relatively low number. It was really that Louisiana longleaf habitat was pretty
Right.
bad for them.
Mm-hmm.
So those first couple days, and then around the Mississippi, all that water, and kind of that long grass type stuff.
Right.
But I think, the winning number was 9 over the course of the week, which is not all that many relative to what we've seen in the Pine Barrens. So
nine that had embedded, or the
no,
okay,
9 that I
just
pulled off
on
of
you.
myself,
Okay.
0 properly attached.
Oh, that's good. Okay.
Yeah.
Yeah.
Yeah, absolutely.
Did you take any precautions, any poison sprays, or
Absolutely.
techniques to try to keep them off?
That was despite putting duct taping our shoes to our pants.
Oh wow.
Yeah, that did nothing for this. Or who knows? Maybe if I didn't do that, then it would have been even worse.
You'd have 9 in your ankles.
Yeah, absolutely right. And it did concentrate the chiggers into my ankles, and maybe that was part of the blistering. I think is what it actually was. Is that the concentration within small areas. So I had multiple bites within like the size of less than a penny, right? I would have 5 or 6 bites within that size. And I think that's what caused that localized systemic reaction to really blow out. So yeah, it was, that was exciting.
Yeah.
We saw essentially nothing in the Louisiana longleaf pine other than ticks and chiggers.
See, if you were a birdo, you'd have something to see.
Yeah,
I'm sure.
maybe there'd be something to speak of. I would say, so we were in habitat because that's what you can do, right? You can't pursue threatened or endangered animals. So we were in habitat where something that would fall in that categorization lives. That would seem appealing given our various lists and proclivities and things. And did not see any.
Yeah.
Humorously, you then had provided information later on in the week that could have made things. It could have made things a little bit awkward, but it wound up not coming to pass. So that was fine. But and I don't know that we were in the same place, but that was interesting. The next day was really concentrated on canebrake timber stuff. Brandon and Owen got their satisfaction, at least somewhat, with a little limited video. So that put the pressure on for the next day. And we went to a different area. They're a little bit tough in that area. And as I say, being so inland, it wasn't what I would really be looking for a lifer canebrake form. So we actually met up with Zachary Gray, who was on the podcast a couple months ago, down in the New Orleans area. And Zachary, we went to a couple different areas, each interesting in their own right. And he spotted this, gravid female that was posted up in leaf litter. Really beautiful, very placid snake, although it was definitely giving us the Varanus vibe in the sense that it was very perceptive to our presence. Didn't move, didn't rattle, we were getting a little bit of sort of blood python-esque eye movement. No, not even, just the eye movement.
Oh, the eye movement. Okay.
Yeah, where it definitely was perceptive of us. But
thought its camouflage was still working because you hadn't grabbed it or something.
Absolutely. And it took a bunch of pictures, literally was swarmed by mosquitoes, was taking all the zen that I had. I think the satisfaction of the moment was enough to carry me through literally hundreds, if not thousands, of mosquitoes all over my face and my hands.
Oh, God.
Just all simultaneously. That was it. Actually, it was funny. So when got up from the photo session, Eric remarked that my face looked like Will Smith in the movie Hitch, and that he was very concerned that I was about to pop into a systemic reaction.
Right.
Totally blow out. It took about 10 minutes, and it actually went down rather than exacerbating, which is good.
Yeah. Oh my goodness. Yeah, you are a dedicated herp photographer. Let nobody doubt that. I would be like, get me out of here.
Well, you got to just hold still, take the bites. We'll
I got my voucher. I'm out of here.
Absolutely. My bus pass, yes.
Yeah.
Indeed. The bus pass was very much at risk. So we left it alone and came back 5 minutes later, and it was gone. So it was doing the Baranid thing where it was like, I know what, I'm going to hold my position. And the second they give me an opportunity to go, I will. But it was really cool to see the firm indentation from where it had been roosted down. It was very cool.
That's cool.
Went back to Mississippi, had a solid day in the longleaf there. Again, few snakes. We did see a young, probably a yearling gopher tortoise, which is very cool. Yeah. Great thing about that habitat. Cruised the scarlet snake, and we did again with Zachary, kind of just him having such a keen eye for the spots within particular localities. Put us on a cottonmouth. That was my lifer western.
Nice.
So that was really cool. And then cruised with Owen. Wasn't feeling well that evening. Had a migraine coming on, which I can understand after all I would put him through. I wouldn't say anyone called this trip super enjoyable, but we did then turn up the local Southern Copperhead as well. So that was really cool.
Nice. Owen does need a few Schmitty tears because he missed out on the Grand Canyon walk. So
yeah, absolutely.
Yeah.
Well, he got some of them here. He didn't cry, but he got some.
Very cool.
But the 3 pit vipers for the area, well, theoretically, I think what used to be called Western pygmies, but I think if you really want a Western form, you'd probably go further to the west, more like Oklahoma. We missed out on those. I think my understanding is that the feral hogs have really done a number on all the Cistrus that were in that area. So very rare to find them. So that didn't come off, but that was so at this point that would be so unexpected that I didn't even count it on the list as something that maybe find.
Right.
But yeah, great trip.
Cool.
Lots of
good times
where you have water. Great time. Yeah, lots of Southern hospitality and
Eric said Eric was the DJ, so
he was. Yeah, that was interesting. Yeah, it was good.
Yeah,
certainly. I don't know that Nipper would have approved, but we had.
Well, it was good music.
Absolutely.
Very cool. Well, yeah, we'll have to hear all the details here soon. We'll either
maybe they'll
get you guys on here or they'll get something going on. Yeah.
Yeah, absolutely.
So there's plenty of podcasts to get on there for. All right. Well, our guest may have something to say about chigger bites or mosquito. I don't know if his expertise translates over there, but welcome to the show, Samuel Kerwin. Thanks for being here.
Yeah, thank you so much for having me. It's a pleasure.
Yeah, looking forward to this. So very cool. I don't know. I was hanging out with an ant guy the other week, and he said that carpenter ants have that their venom has the same severity, or what's the word I'm looking for?
Just like toxicity.
Yeah. LD50. Same LD50 as a Western diamondback rattlesnake venom.
It would not surprise me. We always say, like, in toxicology, like the golden rule is dose makes the poison.
Right.
Like at a certain amount, like water, it becomes dangerous.
Right.
And there's so much similarities when we talk about like venoms across the animal kingdom. Bees have incredible amounts of similarities. Like I think something that we always talk about whenever someone wants to get into the venomous field is like, are you allergic to bees? If so, you should probably take caution.
Right.
You might be a little bit more
extra
sensitive.
Caution. Yeah.
Yeah. Just a red flag, so to speak. So the insect world is daunting to I think a lot of the snake venom people. There's so much more variety when it comes to just everything. Every layer of the insect world is just crazy to some of us. But it's really
incredible, cool. The diversity out there, and the fact that different chigger will cause your whole leg to large bumps to come, things like that. It's crazy. But, all right. Well, excited to talk venom with you. How do you fit into this whole thing, or how did you get into herpetology?
Oh man. I always make the joke where I say poor parenting, where it's just like, you start doing something as a kid and your parents probably don't shut it down soon enough, like wandering around the desert looking for critters. And they're like, oh, he's not going to find anything. And then like a 12-year-old finds a snake. And then, they probably should have stopped the kid from digging around in bushes out in the Sonora Desert, but whatever. It just kind of snowballed into this. So grew up outside just doing stuff. I think this is a similar story to so many people in this field.
I was saying, are you telling my story or yours?
Yeah, right. I mean, I think everyone can connect to that story of just like a kid that wanted to start flip over rocks, and then they see something that they got really into. And I think I had a moment really early on where I was like, wow, this thing can kill me. Why? Right. Like how, like this thing is just a little snake sitting on the ground. Like, why could it do this? And so from a very young age, I was super into just the idea of, venomous snakes in general and like what they could do and why they could do it. So that kind of got me started. And here I am talking about it.
Very
so
cool.
It's pretty fun. Yeah.
Yeah. Where did you grow up in the Sonoran Desert?
So I kind of bounced. I had family both in Illinois and Arizona. Like if you go to Phoenix, you'll notice that there's a lot of like Italian Chicago style restaurants. There's a ton of Chicago, like people going down there. It's like a huge trade back and forth. So I would be in Illinois catching snapper turtles, and then I would go down to Arizona in the summers and find snakes. And I was mostly around Phoenix for the vast majority. And then I actually ended up doing my undergrad at Arizona State, which allowed me to just be there in the desert, living. And it's just awesome. Just love
and Arizona State's in Tucson, right? Or is that?
Yes. Yeah. And it's a crazy place. It's got like 5 different campuses. I actually was like on the far eastern side of the valley. So kind of closer-ish to like Mesa area.
Okay.
Yeah.
Okay. So gotcha. Very cool. Yeah.
Yeah. U of A is Tucson.
U of A is Tucson. Okay.
Oh, yeah.
I get the two confused. Now, I will say that you get them confused as well because I listened to your interview on Snake Talk and you mixed
up.
Yeah.
No, I do it all the time. I do it all the time.
So, I'll get a little forgiveness here. Right? Yeah, we used to have. I'm from northern Utah, and we'd go down. We'd have a couple of water polo tournaments down at in Tucson at U of A. Was that what it was?
Yes. Yeah.
Arizona State's up a little further north. Okay, cool. Were you like? I mean, just kind of general naturalist and had kind of a proclivity to snakes, or did you kind of consider yourself like a reptile guy? Like that was your main, or venomous guy, I guess.
Oh man, it was hard. I can't say like as a young kid, I was ever like a venomous kid. Like just because, anytime you're working at facilities, like I spent a lot of time working at the Phoenix Herpetological Sanctuary down there. And they're not going to let like a 15-year-old work in the venom room. It's a big liability. And so I was very much like, I am like one of the snake volunteers. Like that was my whole thing. But I always like had the goal of just like, I want to work in that venom room. I want to like, actually, have that as the role. But I was always like, really into the snakes in general. Even when like, every once in a while that place is a huge crocodilian collection.
Yeah.
And every once in a while you'd have to move a croc from pen to pen and they just need bodies to help like move this thing. And like, that was super cool. It was an incredible experience.
Yeah.
And at the end of the day, I was like, I'm just going to go back in the snake room, that type of thing. So definitely, the snakes with the direction of the venomous from as far as I can remember.
Yeah, very cool. Well, that's a good place to get your, kind of cut your teeth and get some experience with the diversity and things. Did they have, so along with their crocodilian collection, did they have a pretty good extensive, like, exotic collection of snakes, or was it mostly like natives?
Yeah, they had a huge collection. The facility started early in the 2000s as a home for, just like anything taken in by Arizona Game and Fish or other state wildlife departments. And they very quickly realized that it's like, oh, it's not just iguanas and like sulcata tortoises. Yeah. It's actually like, king cobras in people's apartments in New York and like puff adders in, LA. So their collection grew rapidly. And I think while I was there, it was about 250 venomous, and it was, I think, 115 species, with the vast majority being exotics.
Okay. And where did they get their funding? Like, how do they fund the place?
Lots of state money was one of the big ones. I mean, they worked so closely with Arizona Game and Fish.
Right.
If there was a huge case, they would often hold on to the animals while the trial's undergoing, just to like have a repository. And so they were really, really big partners with Arizona Game and Fish. They had a lot of private donors as well, people that just wanted to support the facility. Tons and tons of like different kind of varied sources.
That's cool. Yeah. Nice. Did you have any projects you kind of considered your own, or that you really were excited about down there?
I think back when I was doing my undergrad, not really. At least at the facility, there was a couple I managed to squeeze in some school projects. Like I have one of my freshman biology classes, you had to design your own experiment with a bunch of your lab mates. And I somehow, amazingly, I still don't know how they agreed to this. I was like, let's do a defensive behavior study between Western Diamondbacks and speckled rattlesnakes in the Phoenix Valley, and see does one rely on crypsis more than the other? Because the hypothesis is well, speckled rattlesnakes are more reliant upon crypsis. They're not going to rattle, or the distance that you can get to them will be shorter before they start rattling. And sure enough, the data supported it. And so that was really cool to be able to include some projects and to get some non-snake people into that. I still don't know why they agreed to do that project. Bunch of 18-year-old, freshman biologists in college doing crazy shit. But
that's cool though. Yeah. I love that kind of attitude though, put a little more research into your things just so you can say conclusively, we've got so many of those, I don't know, old wives' tales in herpetoculture that it's it's nice to have some actual data to support, one way or another. Yeah. That's cool. Even if it seems obvious, of course a speck's not going to rattle as much as a Western Diamondback, but it's cool to actually have the data to prove that instead of
and especially I think in a field like reptiles in general, which I think we'd be hard pressed to find a field that has more misinformation circulating. Right. So getting to put some numbers on a page feels good every once in a while, for sure.
Right. I think one of my favorites that kind of goes around is the idea that baby rattlesnakes are more dangerous than adults because they can't control the venom output or whatever. And it's talk about that one. I'm sure you got some information there.
That one's really interesting. I think every piece of misinformation has a kernel of truth at the very core, and then it gets built up. And, my advisor, Dr. Mackesy, is one of he's got probably more papers on this than the vast majority of researchers in the field. And so him and I make jokes about this all the time. But the kernel of truth is that we see venom phenotypes shifting in a lot of different species from juvenile to the adult stage. The caveat to that is that the volume aspect of the venom gland is an exponentially increasing factor, right? So as a snake grows, the volume of venom it contains is exponentially increasing. So even if the venom of a juvenile is 3 times as strong, 5 times as strong, I would much rather take a juvenile bite than an adult. A juvenile might give me 10 microliters of venom. An adult might give me 500 microliters of venom.
Okay.
So the volumes definitely balance each other out, even if toxicity doesn't.
Right. That's cool. So that's I wasn't sure on volumes. That was going to be the question for you there. So I'm glad you put that out there. And with that, what am I trying to say here? Replenishing the venom stores. Approximately how long does that take? And is it do you have an idea on that?
Yeah, so it is the short answer is 2-ish weeks.
Okay.
But it's 2-ish weeks if all of the venom has been taken out. So if we're doing
totally
a very manual extraction, we're actually compressing the venom glands with our fingers. We're going to see roughly a 2.5. We say about 16 days is typically when we see the full replenishment of the venom gland. And we actually measured that using transcriptomics. So we can actually see when do the genes coding for venom start being read, and then when do they stop being read? So when do the proteins of venom stop being produced? And we see that typically is around 16 days is the max. Now, if a snake in the wild were to bite something, it's not going to use all of its venom. I think some estimates from Dr. Bill Hayes out of Loma Linda University estimate a third-ish of their venom yield might get used in a single predatory bite. Varies wildly, obviously. And so that will decrease the amount of replenishment time that we see. So it's pretty linear with venom amount expelled equals time to replenishment.
Right. Does how long they've gone without a meal play any role in volume injected?
It may.
Is there just too many unknowns out there that, these kind of things?
There's a lot of unknowns. I actually, when Dr. Hayes first presented some of this, just about like the venom expenditure studies he was doing, I asked him, how do you measure that? Because first off, you have to get them to bite a mouse.
Right.
Well, how do you measure the amount of venom that was injected into the mouse? And his explanation for that was not what I expected.
Really? Yeah.
So we can detect proteins using a bunch of different methods, and we can actually take like a sample of venom that can tell you how much of that venom is a very specific protein. Mm-hmm. He does the same thing, but he does it on the mouse level. So he can actually wash off any venom that's on the surface of the mouse. Mm-hmm. And then he can, what he calls homogenizing the mouse, which is to make all the same. So he puts it in a blender, and then he does that where he samples it. And then he can say, here's the proportion of the mass that was venom. And then give you an estimate of this is how much venom was injected into that animal with pretty good accuracy, and enough to at least give him some good numbers and allow him to make some inferences. So.
Okay, so if he wants volunteers for his studies, don't volunteer for that study.
But I mean,
that's impressive.
Then you have to consider, the follow-ups to that is, was it one fang? Was it two fangs? What was the duration of the jaws on the actual animal before the snake released? Was it a holding bite? So it sounds like such a rabbit hole to go down as a researcher that I asked him about it, thought it was cool, and I was like, can't wait to see you present on it in a couple of years.
We'll let you cover that field. Yeah. That's cool. Yeah. What an interesting thing. Yeah, I was wondering how they'd measure that. That's insane.
It's a big graphic for sure.
Yeah.
Do you know, is there anything, I certainly I've heard the discourse around it, and I don't, recall which way exactly it goes, but the idea sort of the effects on humans being variable based on the intended normative prey item for a given species. So I think the suggestion was that there is a certain effect when when the anticipated prey is a mammal versus an amphibian, and that sometimes it means if it's essentially totally intended for a totally foreign animal, that might mean it's no impact, or it actually might mean that it's worse. So that things that are eating amphiumas or frogs or whatever might actually be worse going into a mammal than something designed to impact a mammal.
Yeah. Oh man, that one's tough. I've had a few ideas about this for a long time. They're totally unsubstantiated from a data perspective, but I think there's so much of it that goes into how a bite was initiated, not only from the animal's perspective, but from the human perspective. Did I, put my foot next to a bush and the animal who was waiting in ambush did a predatory bite? Or was it an animal that was not in ambush and, the same situation happened and it was a defensive bite? And so, man, the behavior of a bite is super variable. And then tack onto that all these other factors of what type of animal was it waiting for, what posture was the snake in, prior to the bite, what extremity did the human get bit on? Because, the human factor is crazy as well. I mean, every human reacts totally differently to venoms, and not totally differently, but there's variation within there. Like an Aatrox bite is going to suck for everybody, but the levels at which it sucks are going to be very different. Did you eat your Wheaties that morning? Do you have certain amounts of minerals in your blood that might interact with certain toxins? The layers when it comes to that is dramatically, I think, understated a lot of times. I think as snake people, a lot of us focus on okay, what was the snake doing? But then it's okay, what are the health issues of the human that just got bit? And what's their mass? What's their BMI? What's all these other things that go into it? And the MDs that we work with on occasion often stress their frustration at how variations exist all the time. It's just snake bite just makes it a little more complicated and exemplifies some of that variation.
Right.
So,
I'd seen some research that tested, defensive bites versus predatory bites, and it seemed like about half of the defensive bites had venom injection, whereas 100% of the predatory that was kind of what my takeaway from it. Can you confirm or deny those that remembrance?
Yeah. I mean, I think that was also a study that was done by Bill Hayes doing like that whole strike behavior stuff.
Right.
I think that often gets thrown into the realm of like, oh, it was a dry bite type of thing. And I truly don't think there's anything called like a dry bite ever. I think if it's a venomous snake,
there's bite, you're getting something.
Yeah. And it could be a small enough amount to cause a blood blister or like a minor rash. But I think if you're going to get hit with fangs that have like a residual amount of toxins on there, you're going to suffer something. The only reason I say that is because I have poked myself on fangs before that, did not pass through an animal and were just falling out of the mouth while they're feeding. I've also like poked myself with needles that have like very, very dilute amounts of venom in them and had a blister form. Didn't even hit the plunger, just poked myself with what was residual on the tip of the fang or tip of the needle. I think Bill's work definitely gets at that like venom can be controlled very well. We see it get controlled. We know that venom is a valuable resource. Like it is energetically costly to produce, 50 milligrams of protein in a venom gland and have it stored there for a significant amount of time. And it's more expensive if you consider that is their tool to get more energy. So they're unwilling to use that. And I to people that are afraid of snakes, I think that is a great line of dialogue to use with them. Just describing that it's
they
like,
don't want to use it on you.
Yeah, it's a tool. I mean, just like anyone who's out in the wilderness, if they're like hunting squirrels with the 22, they probably don't want to use all their ammo on a bear. They probably just want to try to scare the bear off before they ever try to waste, their resource to survive and live. So,
yeah. And is there anything known about like the mechanics of a bite, like a maybe defensive bite where they're maybe trying not to use their venom, or is a bite? You know what I mean? Does that make sense?
Yeah. So mechanics in terms of like how the venom is administered and like what choices go into that,
right? Or I mean, are they controlling it at the pumping out of their venom glands, or are they control it, or is it like if the fangs go in, venom's flowing out? Is there control? So, where's the control lie, I guess?
So a lot of the work that my advisor and I, or really my advisor has done, and then I've kind of tacked onto in the past couple of years, is really looking at like the physiology of how his venom is made. And doing that means that we have to, look at a lot of venom glands. And, and so that combined with, like working in a museum where I'm preserving specimens a lot of times, you can very clearly see the musculature that is designed for venom expulsion. And it's all based around like the actual gland itself. And we call it the compressor glandulae muscle. And the compressor glandulae, it doesn't necessarily squeeze, because squeezing is inefficient. What it does instead, and this is something that almost every human is familiar with, is it wrings out. So it actually wraps around and twists in like a torsion motion, just like how you would trying to get the water out of like a wet t-shirt. And it's very, very effective. And it can induce, I think, up to, I might be wrong on this, it's been a while, I think it's upwards of 34-ish PSI within the venom gland. The membrane, like the actual fang itself, because this is also something that I think people assume, is that it's wrapped in that, the fang is wrapped in that sheath. And the sheath will slip up once the fang actually punctures something. I don't think, and I don't see any reason why this would be true, I don't think it actually is the trigger mechanism for venom expulsion. I think it simply allows for easier venom flow, because that membrane does actually cover the external region of the fang. And something that we see when we do venom extractions is if you don't clear that membrane, venom still pools in the membrane. It is just trapped there like a little water balloon. And so then we have to be finicky and try to get it around that little water balloon. But venoms can still flow without that membrane being lifted or removed from the actual fang itself.
So is the membrane resistant to puncture from the fang?
Yes, in a way. I mean, it's super, super elastic. It's very similar to like, any oral like tissue surface that you have, especially like cheeks, where yes, you bite them on occasion, but very, very elastic tissue. And so we often see that it can stretch very, very far, and we can actually slip it back, and then it folds up very, very neatly up in the roof of the mouth when you see those really cool like fang shots that people take all the time.
Right. Very cool. Yeah, this is stuff. I mean, you maybe think about it in passing, but that's really cool to hear the actual, truth behind the thoughts, I guess.
Yeah, absolutely. And while we're there, would you mind going into your master's project a little bit? This fell into the box for me of saying I hadn't even considered it as an issue, which is amazing, right? As a sort of scientific question. It was something I'd never considered, and the answer that you were able to unearth is pretty amazing.
Yeah.
Oh, man, this has been like a really, really cool thing. And I love getting to talk about this because now my research has shifted dramatically. There's a few decades of work that kind of laid a lot of the base level information that we went on for my master's work. A few individuals from my advisor's lab, especially I think it was like late 90s, it was 1998 is as far as back as some of this work goes, where my advisor was actually teaching an anatomy and physiology class, like a human anatomy and physiology class, which he doesn't like teaching about the human stuff, but he has to every once in a while as a professor. And he made this observation when he was doing some like cell imaging and just like some histology, some cell staining. And he noticed that there are cells within venom glands that look remarkably similar to the gastrointestinal system of humans, where we have these cells in our stomach that are actually producing acid, which allows for, digestion. It's a huge part of it. And so he made that observation and he kind of just put it in the back pocket. It's like, cool. He had a student that came in, looked at it a little bit more, and he's like, okay, these are definitely acid producing cells of some kind. That's about it. That's all they got at that point. In 2006, they had another big study on it where they did the same thing. They, pulled everything apart. They looked at all the stains and they wanted to see, okay, if it is acid secreting, is it the same type of acid secreting that we see in humans? Is it an actual parietal cell from a human GI tract just now in a snake venom gland? And what they saw suggested, no, it's not the same thing. It's a little bit different. It's maybe a little bit more, not necessarily inefficient, but it's not identical. It definitely has a reduced functionality in terms of acid secretion. And then that begs the question, why on earth are you pumping acid into a venom gland? Like what is going on here? Why? There's so many more questions are popping up at this point. Yeah. And it all comes down to another observation that my advisor made. He was just on a whim. He took the pH of some venom that he got from some rattlesnakes and found, okay, the pH of these two rattlesnakes, different species, is the same. It's like 5.5, so slightly on the acidic level. He contacted a collaborator that at the time was working on Bitis and like a bunch of gaboons and puff adders. And he asked, next time you take venom, just take a pH reading, let me know what you get. And the advisor was like, his collaborator, was like, I think it's around 5.5, like 5.6. He's like, well, that's super strange. Like you have two, groups of animals, two different continents that are now having the exact same physiological number coming out of an excreted like venom profile. Again, why? Let's dive into this further. Yeah. And we started working with the genetics lab down in University of Texas, Arlington, the Dr. Todd Casto in his little lab. And they do a lot of evolutionary genetics and transcriptomics. And they found that during that venom production cycle, yes, venom is upregulated. We see tons of venom proteins being produced. We have all these venom genes being transcribed and everything like that. But there's a lot of other things going on in the background. Upon venom excretion, there's roughly like 2,500 different genes that get turned on or differentially regulated within a 24-hour period. And it's a huge shift. And you can actually watch the cells in the venom gland expand and grow to accommodate all of the protein production. The images are insane from some of these studies. Wow. And among those are all of these genes that regulate for these acid secreting pumps. And so that study was completed in 2020, and it led us directly into my master's program, which I began in 2021, which is, let's solidify why they do this. Let's try to figure out, like, okay, what are the actual mechanics of this and what is it doing for the venom? And then let's also try to turn them off and watch what happens. Like, can we turn these off in live snakes and stop this acid production so that we can really put a nail in the coffin and support this idea of, why potentially the pH is being decreased. What we found was two things. Number one, as soon as venom is put in that higher pH, because it's at that acidic pH when you pull it, put it in a little vial. So as soon as you put it artificially at a higher pH, things start destroying themselves very quickly. And this is mostly due to a couple toxins being protein targeting toxins. So they're very good at destroying protein. Well, venom is also protein. And so they just start tearing each other apart. It's a bloodbath on a biochemical level. And it just results in a really, really inefficient venom at the end of the day. If you let it sit in that higher pH and then you try to test it again, you see dramatically reduced toxicity across the board.
How high are we talking? What kind of pH does it start to degrade or act on itself, I guess?
So we put it, we were doing this at physiological pH of around 7.4. That's really where we wanted to see. That's a super common pH in the biological world. Almost every animal has that as like their blood pH or tissue pH, unless it's like the stomach or other organs. So we saw that decreased functionality, huge red flag. Okay, maybe it's just protective. Maybe that's all it is. Maybe it's just trying to prevent the toxins from degrading. These are animals that don't use venom very often. Like some of these snakes are going months and months and months without food. And they also don't live in the most stable environments. We talk about like biochemical stability. Like for us to keep drugs, we have to put them in freezers. Right? These guys are living in the Sonora Desert, where it's probably like 109 right now. And so it goes on and on and on. And I'm like, okay, this is starting to become a much clearer picture. The next big question was why, like other than protection, now you have an acidic venom that's sitting in this tube and it's not really working. Like everything's super inefficient. The proteins aren't folded properly. They're not destroying themselves, which means that they're not active. How does it get turned on? And this was like the kind of aha moment for this whole study was, the pH is 7.4. That's when they work. So our bodies provide the perfect environment for these toxins to fold properly and kick on and start, going to town. And this kind of was this really, really cool moment of like, wow, these animals are using our own physiology against us and their own prey's physiology against them. And it's such an elegant solution to a really layered problem of toxin production next to your brain over long periods of time. And it's really, really cool. And there's so much more to learn about these things. So much more.
That's incredible. Yeah.
Absolutely. Did you guys wind up seeing did you have the capacity to evaluate reducing or making more basic the venom within the snakes?
Yeah, that was probably the toughest part of the entire study was like, okay, we know what these kind of do. Let's see if we can turn them off in a live snake during that venom production cycle that, they're usually super active. And we used a drug that actually targets these very, very specific pumps. One of the great things about working in research is that you find a bunch of weird drugs that are just like out in the middle of nowhere. Like they're not pharmaceuticals. They may have been tried to be used as, some sort of drug product in clinical trials, and then they didn't make it, but they're still for sale. Yeah. We found one that does work on these things. And we tried, I think, 4 or 5 different methodologies that were as like uninvasive as possible to try to not mess with anything else. And it just kind of led to headaches, as most research does at times. And so we ended up just directly injecting the drug into the venom gland. And we did have some success with that. We actually had venom pH increasing significantly across all of our trial animals up to about 6.5, 6.7 was about as high as we were able to get it. And we saw pretty dramatically reduced venom functionality at that point. But it was a different type of impaired functionality than what we saw with just the artificially improved pH or artificially increased pH, where there's definitely another role going on. It's not just pump acid into gland. It's more so probably pump acid and help venom be produced type of thing. So there's layered functionalities to this in any biological system. If you mess with one thing, other things are going to inevitably start going wrong. And so,
Right.
That's definitely part of it. But it was a really cool look at what a lot of us, I think, forget about when we talk about venomous snakes. We always talk about the venom itself, but man, the way that they produce it is just awesome.
Right. So did the LD50 dose go up with those 6.5 animals, or?
Yeah. And so we never did those LD50s just because they're pretty variable in general.
Okay.
So what we actually do is we can assess specific toxin families. So we can, if I want to assess snake venom metalloproteinases, which are really, really common in snakes that cause tissue damage, and I only want to assess the functionality of those, I can actually do a test in like a test tube that will directly quantify that one toxin family. And so it's a bit more specific and allowed us to pinpoint, okay, which toxins are being more negatively impacted by this increased pH versus others. And there are some toxins that don't care, and which is really interesting because those happen to be the same toxins that cause prolonged issues in humans. And they're not just the ones that hit you hard up front. They're the ones that cause issues over the course of like a week or two. And so there's a neat correlation there with the stability of toxins and the effects that we see in humans.
So the ones that are stable both at low and high pH, those are the ones that kind of remain active for a longer period of time.
Yes. Yeah.
Wow.
Especially some toxins that have caused some havoc in the US in terms of like the medical field that have kind of encouraged a couple new antivenoms to be introduced. We're definitely up there. There's a lot of reports of human patients that, especially I think pre-2018, it was a low incidence rate, but it was enough where patients would be cleared from the hospital, discharged. They would have been treated with antivenom, and they will have no, severe symptoms at the time. And a few days later, they would come back with a recurrence of this coagulopathy or disruptive, like hemostatic system where their blood couldn't clot or was either clotting too much.
Right.
And that was a big issue. It meant that the antivenom was leaving the body before the toxins were actually being degraded or metabolized.
Right.
And because it's coagulopathy, we can narrow that symptom down to two major toxin families that we refer to as serine proteases. And when we were doing our study, the serine proteases were two toxin families that did not care about pH changes. They are hyperstable enzymes. They're incredible at what they do. And it just makes sense that, of course, those are the ones sticking around causing issues. They're incredibly stable, even in the lab when we try to mess with them. So really, really cool correlation between those two just kind of observations.
So how does their effector function differ in the venom gland versus when it's in a person? How does the venom gland is protected from these serine proteases?
So a lot of it is due to targets. So if there's really two ways to stop an enzyme. You can either destroy the enzyme itself, causing it to fold improperly or actually tear it apart, or you can remove its target, right? If you take away the substrate that toxin acts upon, then it can't do anything. So these are toxins that primarily target things that are in the blood and in the hemostatic system themselves. So inside the venom gland, there's no circulation. It's just a actual pocket of space. So there's nothing for them to act upon within that area.
Gotcha. And do you notice a more higher presence of these proteinases in like more advanced venomous snake species versus those that just kind of have a more of a primitive venom duct? Or is that still unknown?
It's an interesting. There's not a really good set pattern on where we typically see, these proteases starting to come into play, or at least the serine proteinases. They are oftentimes, if they are present in a venom, they may not be one of the predominant toxins. They may be present in a significant amount, but to me, a significant amount in a venom is 10%. And so you could have other proteins that are present in 25, 30% that are the predominant toxin family within that species. And that's typically where we see variation on those very, very dominant toxins. There's a lot of other venoms out there where you've got 50, 60 components, and 45 of them may do something, but they may not be as important as like the 3 or 4 very, very big main characters.
Okay. So in a typical venom, you're seeing probably upward of 50 or so different components or active ingredients, if you will.
Yeah, I'll say with vipers, definitely.
Okay. Wow.
Vipers definitely are they probably reign supreme in terms of venom complexity, just generally. There's definitely some vipers that break the rule and decide that they want to be like elapids, but
and just for comparison, what do you see in an elapid typically?
Elapids will actually typically they'll really be dominated by a couple, like 2 or 3 super major toxin families, and sometimes it's only one major toxin family. And we might see variations within the different isoforms or kind of flavors of that toxin within that one species, but they have very, very simple venoms. One of the ways that we visualize venoms is using something called gel electrophoresis, where we can actually separate the venom by all the components based on their actual size. And what we see with vipers is you have this almost looks like a ladder where you've got bands just appearing all over the place with varying intensity. Some are super dark, some are very, very light and faint. And if you do the same thing with an elapid, a lot of times what you'll get is one huge band at the bottom and then a few very, very, very light ones just kind of scattered around. But it's nowhere near what we see in the vast majority of vipers, especially rattlesnakes here in the U.S.
Okay. Interesting. That's cool. We've got the most advanced venomous snake venom in the U.S. or in the New World, I suppose.
The acidification, right, being maintained in that state, that it does apply to the enzymatic toxins, right? So we're talking principally about vipers, that this is not an impact that we're seeing in elapids. Is that fair?
Yeah, I mean, there's more variation in the venom pH when we get to elapids. There was a paper that came out about spitting cobras, well, not just spitting cobras, a lot of cobras, and their different venom pHs. And it definitely had a wider range. I think the highest I saw there was like 6.2, which is something that you would never really see in a lot of vipers. And that's not a huge jump, but pH is a logarithmic scale. So, a change of 0.5 is actually very, very significant in terms of what the pH value is inferring. And one of the things I noticed is that the elapids that have that very consistent like pH, the viper level pH of 5.5, 5.6 are almost always spitting cobras that do have a large amount of enzymatic toxins. So it's almost like because they're going to use that enzymatic toxin, they also have to do that viper thing of having acidification in there.
Interesting. So, what about a viper wannabe elapid, like a death adder or something? Do they have?
I don't know. I don't know.
Could lifestyle play a role where they're more ambush predators versus like the taipan or something that's chasing down its prey?
I have actually asked a few researchers down at the University of Queensland, where Dr. Brian Fry is, to next time you guys take venom, or if you do ever take venom, just give me a pH reading. I mean, it would be interesting to know. I mean, Australia is an outlier for a lot of different reasons, and it would be interesting to see if they follow their trend of just being kind of weird from an animal perspective. But I'm not sure. Australia is kind of a black box for this. There might be some literature out there, but I have not seen it, if so.
Interesting. You'd think with so many species that are venomous down there and so many elapids that have varying degrees of, human impact, they would have a better idea.
Yeah. I mean, they have tons and tons of research. Right. Their knowledge level is very extensive. I will say the acidification stuff was it came out of the blue, and it's a bit niche, and it's more of, it definitely falls in terms of our researching categories into a very kind of almost ecological, like biochemical ecology or biochemical physiology,
right?
More than, you know, the human impact side. Because while it's cool to have those correlations and observations, I think an MD is going to look at that and be like, I'm not going to acidify the blood of my patient to try to cure him.
Yeah, has no applicable avenue.
He's like, this is cool, but whatever.
But from an evolutionary perspective, that's really important and interesting. Yeah, for sure. Okay, that makes sense. Yeah, I guess we won't bag on the Australians, especially here. That's one of my favorite places. So gotta love the Aussies. Yeah.
When you've talked about the master's project before, you mentioned there being hiccups along the way, and I was just wondering if you could talk a little bit about sort of what does the scientific research process look like, and what are the sort of unexpected challenges and trials and tribulations you run into with that? I know Justin can relate, so that's why I think to actually hear about it. And too often, right, I think that doesn't come out. It's focusing on the positive results rather than really getting into the challenges and difficulties you can run into.
Yeah, I think even as researchers, we really want to focus on the positive results sometimes and forget about the headaches.
That's what we publish, right? We don't publish negative data usually.
Gosh, the headaches in a lot of this research are, you know, they're in the scientific in scientific context, they're very human. Where, you know, we just have those moments where we got to like sit down and just put your head between your hands and just kind of like rethink a lot of different strategies. One of the things I was finding early on in like some of my literature review was, has anyone like tried to alter the venom gland environment during like in a live animal? And the short answer is no. I looked for a really long time and I was kind of crestfallen that I thought it would be at a dead end. And I kind of mentioned we wanted to be as uninvasive as possible, but uninvasive as possible still means kind of invasive, especially for this context. Especially when I had the experimental design at the time. It was very dumb. When it worked out, it was great. It was super smart after it worked. But when I was first trying to develop it, we had this idea of, you know, you have a snake with two venom glands, and each venom gland is totally separate, biologically separate. They're physiologically separate. They can respond to different signals. They have their own venom production. They're not sharing venom at all. Can you give one a treatment and the other one a placebo in the same animal reduce the animal to animal variation using that
internal control?
And you know, at the kind of the front end of that experiment, I was like, oh, this is so cool. And at the middle part of the experiment, I was like, oh my God, this is so torture, because we have to give a drug to each side and not impact the other one, and find a way to administer the drug that keeps it in that specific area. And so we tried something at first called osmotic pumps. These look like little pills, and it's a gel membrane with a little like balloon inside and then a little cap on the end. And you can fill it with a certain substance, and the gel will take in water from the surrounding tissue that it's been placed in and put pressure on the balloon. And the cap will ensure that you are only pushing out a certain amount of your drug at a certain rate for as long as it will allow. And so it's a really, really cool little piece of technology that works really, really well in mammals.
Right? Yeah.
Yes, all Zs. Yeah.
We use them all the time.
Yeah, they're amazing, and they work great in mammals. And you know, I thought, okay, this is going to be a cinch. It's going to be awesome. It's going to work perfectly. I found out very quickly that when they're administered subdermally, just above the venom gland of a snake, they typically don't have enough just ambient osmotic pressure to actually push any drug out. So what we were resulting is we would take the venom, or take the pumps out of the dermis, and we would find that there is a full reservoir there. And that was like after a week. And so we threw that idea out, and we started trying to do, you know, a bunch of different things. We tried priming the venom glands, because the last thing we want to do is puncture them. These snakes are really, really resilient, and we never saw any adverse effects from doing this, but we ended up having to, in the end, is actually giving an injection directly into the main venom gland luminal space, and then letting it go from there. And we would do 2 injections per like venom production cycle, and that was pretty good in terms of like actually getting some pretty good results in the end.
Interesting. What did you ever consider anything like RNAi or anything like that, where you're blocking the genetic or the transcript?
Yeah. We did. We thought about that a little bit. We wanted it to be as well. I think the very core issue was like, this is a master's project. We got 2 years.
Yeah.
Let's see what we could do.
Yeah. If it doesn't work quick, get rid of it.
Yeah. And it really was a shot in the dark. Like, yes, we had a good idea that this was, what was going on and that we could potentially inhibit it. But I think we really, at the end, were just kind of hoping that it might show us some results. Now with, the confirmation that we have, I don't think any of us would hesitate to try something a little bit more, I guess, intense as trying inhibitory RNA and see if we could block the transcript itself. We actually, for a moment, discussed doing gene knockout snakes, where you actually knock out the whole gene for it. That has its own complexities of you have to do embryos and then grow them,
right?
Which is a bit of a pain. Yeah. That's more headaches that I want to take on. Yeah,
yeah.
But yeah, I mean, I think now we're interested in potentially doing more in the future, but maybe when our plate is less full, full, which might be never. But
gotta pick that low hanging fruit first, right? Before you get into how about a injection through the fang?
Through the fang? Yeah. We tried that as well. So we didn't try it. We talked about it extensively.
Yeah.
I got talked out of the idea by my advisor, because he's been studying venom gland physiology and morphology for a lot longer than I have.
Right.
He might not like if I say this, but before I was even born, he was studying it. And we talk about snakes having this really good big identifiable gland, and yes, it's identifiable. Yes, it's this big pocket of space. But then you go through a little duct that leads into a little tiny accessory gland. And then from there, you have another duct that then leads into the fang. So your injection would have to go through an actual valve into the accessory gland, go through another valve, and then into the main venom gland.
Right.
And maybe it's the right fang. And let's say the snake has two fangs on one side, and one of them is a looser connection than the other one. And you go through one, it doesn't make it. It pulls up at the maxillary bone and doesn't actually go anywhere. So that was a little bit more variation than I think we wanted to try.
Okay.
So we did think about that.
Yeah. Well, that was kind of my next question too. And is when they replace their fangs, how does that work? That's crazy.
Gosh, I don't know. I don't have, I don't have as much information on that. It's something that I've talked about. Just you have a beer or two at a conference, and then you and 3 other scientists start talking about all these different things and just throwing ideas back and forth. And Dr. Bill Hayes and I, once again, were chatting at a conference, and I was like, I think it goes like this. I think they grow in a fang comes in from where they're all in development, and the membrane around that maxillary region grows to accommodate it and accept it. And he's like, no, I think it goes like this. And so we went back and forth on ideas for a while, and then we just settled on we'll have to test it one day and just see, what goes on. I think it's going to take someone that's more of a developmental biologist individual, someone who's used to doing things like
tracing, where
just a ton more in-depth. Say confocal microscopy could probably come into play, looking at tissue growth and how it's growing, things like that. But yeah, something that I'm definitely not equipped to answer in the best way.
No, it's fun to think about those things for sure, and just kind of, somebody with your education and background would have a lot better. If I'm putting it out there, it's simple and stupid. I'm sure. But,
I think anyone who's seen the skull of a snake or has looked very, very closely at how those fangs connect can make a bunch of inferences. You see if you're looking at a maxillary bone, you clearly see 2 ports, and you clearly see that they're connected, but you also see if there's one fang in, it's a very porous connection, and it's bone. It's set in there with bone. But how does that connection grow? When does it grow? What signals it?
Right.
I mean, there's a million questions there that, I always ask. And I think all it needs is a couple observations, and anyone can probably have some good ideas.
Right. Yeah. And I guess one hypothesis can be as good as the next.
Yeah, until you get those numbers, you don't know.
Right. Exactly. Very cool.
Yeah. Man.
Absolutely. So you want to talk a little bit about what you're doing now, and then the whole lab, right? Everyone's getting involved. They talk a little bit about the whole idea of the customized, essentially the viruses in yeast cells being hyper-focused, all that stuff. That's mind-blowing.
Yeah. Oh man. I got to present this stuff at Copark, where obviously you heard it. And I loved getting to present this stuff there because that is a uniquely invested crowd when it comes to, the whole topic of venomous snakebite treatment and, what are the innovations that are coming in the pipeline. I think a room full of, field herpers are going to be very, very attentive to that topic to see, how expensive is it going to be? Is it still the same price as modern anti-venom? Can I have a little flexibility maybe next time I go out? Yeah. There's a big shift. And describe, I kind of have to start from square one, kind of like I did with the talk, to really where antivenom begins. And it begins like using basic rules of immunology way back in the kind of late 1800s with bacterial infections. And bacteria, especially diphtheria, will produce toxins in the actual surrounding environment. And a couple of researchers found out that if you take the liquid off of like a diphtheria culture and you inject that into an animal, then that animal will produce antibodies that protects against the toxin that the bacteria produce. And so the first ever antivenom is not an antivenom, it's an antitoxin against diphtheria and tetanus. Mm-hmm. So it's really cool to see how that gets published. And within 2 years, so another researcher saw that and went like, snake venom, easy jump in logic. And like that to me, like some of the early developments are really, really cool to understand. Now, at the time, they didn't really know that, hey, snakes have different venoms, and they're wildly different to the point where you can't just have a cobra antitoxin and then use it in the U.S., right? Obviously doesn't work like that. But at the time, like they didn't know. And so they were just taking animals that looked similar within a specific region, using them to create this kind of antivenom by taking the venom, introducing it in the large animals, and then just taking that plasma and injecting it into humans. And that was problematic. You can't just inject full animal plasma into human. Their bodies tend to freak out and not do super well. And so a lot of the innovations in antivenom from like 1890s-ish all the way to really like 2016, 2018 were entirely focused on two things. One is making antivenoms that are targeting geographic regions with hyperspecific, not hyperspecificity, but like with more specificity than what normally had been done, and then also making it a lot safer. And like so that humans wouldn't, react and go into anaphylaxis when you're trying to treat a snake bite. And the anaphylaxis part got handled pretty early on. There's a couple techniques that we use to kind of clean up antibodies, and they're now pretty safe. Like the adverse reaction rate is in the single digits in terms of percentages nowadays, especially in the U.S. And honestly, at the end of the day, if like a medical doctor cannot treat anaphylaxis in a hospital, they probably shouldn't be a medical doctor. Like you have every toolkit in the box ready to treat some person going into anaphylaxis right in front of you. And so the moral of the story is, antivenoms work in geographic regions nowadays, and you can, even with a mild adverse reaction, easily treatable and not even significant anymore. Right. So we have safe products that work in whatever space that they're used in for the most part. This is kind of a rough design though, still. Like at the end of the day, it's gotten more specific, it's gotten safer, but you are still taking venom, injecting it into a big animal, pulling off plasma, and just refining it a little bit. That's it. It's the same formula that was first used in the 1800s. Right. It's not like we've really changed the game. We've just gotten a little better at playing it. And we typically refer to this as kind of like a shotgun approach, right? Where you're not targeting individual toxins. Like I talked before, you can have these hyper complex toxins or hyper complex venom profiles within, especially rattlesnakes here in the US. Right. And only 3 or 4 of those might be main characters. You're going to have a bunch of stuff that's not necessarily toxic. They could be, other peptides that are signaling things. It could be like cellular debris that's produced in the actual venom gland and just pushed out. It's not even part of the venom.
Just foreign. Yeah.
Yeah.
Yeah. Your body's going to mount an immune response to anything foreign. So
yeah. And there's a couple people that have done some work on, how can we make modern antivenoms, like make slight adjustments to even account for that shotgun effect. And CroFab, which is, been in the US like medical system since around the 2000s, like did a pretty good job at that. They were one of the first ones to really do this. They did affinity profiling or affinity purification, where they actually have a column that's lined with venom toxins, like all the toxins that they really want to target. And they would take this antivenom or plasma straight from the animals, and they would feed it through the column. And as it goes down the column, all the antibodies that are actually attacking the targets will stay in that lined column. And then they can then remove those and have a much cleaner product. Mm-hmm. That is typically why CroFab is a much more expensive antivenom. Mm-hmm. So it's not just the U.S. medical system, although that does play a big role in the price. Like it definitely is a good product, but it's still, had the same thing. Like you have a geographic venom that is supposed to cover, how many species in the U.S., and it's supposed to cover all of them with pretty good effectiveness. And even with this purification, it's still really, really variable. And on top of that, in a lab setting, for me to ever see really good inhibition of a certain venom, whether I'm doing it in an animal model or I'm doing it in a test tube, I need almost 100 times the mass of antivenom that I would venom. So if I use a milligram of venom, I would need 100 milligrams of antivenom to get anywhere close to trying to inhibit that. And from a biological perspective, especially that biochemical perspective, that is horrendous. That is insane. That is the ratio that you need from one to the other.
Is that just the antibodies binding to the protein? It needs that ratio?
So that's to see a totally inhibited effect. So 100% inhibition of these toxins in a test tube requires you to get up to that much, if not more, for some of these toxin families. And that goes back into the idea of we have venom variation, right? Yes, this is a geographic polyvalent antivenom. It's supposed to cover everything. But let's say you have a certain amount of your percentage of antivenom is targeting those serine proteinases, and you have one species that has a ton of them. Mm-hmm. You're going to need more antivenom to cover that one species. And so, there's so much variability with that. It just adds layers and layers to it. But at the end of the day, it's still the shotgun approach. It's a cleaner shotgun. You've gone from birdshot to buckshot at this point, I guess, but you're still doing a shotgun approach. And, that's cool. It works, but it only works in the US really because we have infrastructure to support it. Right. We can dump 20 something vials in a person. There was an Eastern Diamondback bite in Florida by an influencer. And I think he got 88 vials of antivenom. I think it was earlier this year or late last year.
Yeah.
I just remember watching the whole bite kind of play out on social media. And I was just that is not going to be good. And I think they ended up giving him almost 90 vials. And if each vial is retailing for, $13,000 or $15,000, I think I probably would have let the bite take me at that point. That's just insane. It's astronomical.
Yeah.
And so that goes to highlight, you need that much antivenom for a single bite. That is outrageous.
Yeah.
Because each
one of those influencers, they got the cash, they get.
Gosh.
Oh boy,
man! I would pale to see what that bill was.
Right.
But if each vial of antivenom contains 75 milligrams of crude protein, and that snake bite was probably only 30 milligrams of venom, that's a huge amount. That's a 1 to 300 ratio. Yeah,
right.
It's crazy.
Wow.
And so. There's a need for a shift in how we do things. I think that is kind of this long-winded way to say that. We're at this point it works in the US. We don't think about it as much in the US, but there are other regions where you don't have 88 vials to give to a patient. You have maybe 2. And you better hope that 2 is enough so that they can make it through the night, and maybe you'll give them 1 more.
What about like cell-based synthetic approaches like hybridomas or something that are producing targeted, specific antibodies that work, and then you could kind of mix them in a cocktail depending on, the venom components in the region?
So it's super interesting you bring that up, because that's kind of where like a lot of this is going for the most part.
Okay, cool.
So we, in collaboration with a bunch of different labs, oh my gosh, I think there's 4 countries involved. The vast majority of the labs are in Europe, and the big kind of brainchild of the projects are really in Copenhagen, Denmark. Cool. The Denmark Technical Institute, Denmark Technical University. I think the lab is currently headed by Dr. Andreas Lausten, and this is a antibody development lab. They're not just a snake venom lab. A lot of them have never even seen any of these animals before, and sometimes we'll send them pictures of stuff from our lab, and they're freaking out and they love it. And we love to chat with them at conferences. Yeah. But they really study antibodies, study how antibodies interact with things. Can we create better antibodies? And they reached out to us along in collaboration with a Mexico lab, talking about doing some work with these nanobodies. And nanobodies was kind of a foreign concept to me. I didn't know much about them. And we talk about antibodies being the things that have always been used. And those are those really classic Y structures that we see all over the place. They're probably one of the most abundant antibodies in humans. But not all mammals are created equal in the immunological sense. Camelids, like llamas, alpacas, camels themselves, actually produce things called nanobodies. And so it's the same kind of basic structure where you have those 2 parts of the Y that are binding to different antigens and inhibiting them. And then you have that kind of big stem part. But those 2 little top regions are really small. They're only about 1/4 of the size of what we think about in an IgG or immunoglobulin you find in a human. And so that became really, really ideal for a couple of reasons. But one of the big ones was the whole idea of you're injecting an antivenom into a patient. You're injecting it into the bloodstream. The venom itself is not in the bloodstream. It's, in muscle tissue. It's in lymphatic tissue. And so the antibodies are really good at binding up, anything that's in the blood. But that's why you have to keep redosing antivenom, is because it's you'll have toxins leaking into the blood throughout the course of a bite. The nanobodies are small enough that they have really good distribution qualities, and they can get around to some of the areas that the actual antibodies can't. And so that was a really promising thing. They're also a lot more stable, which means that they have a longer shelf life. So another win for them. Right. And one of the things that this lab did, and I did a lot of reading to kind of catch up to where they were and just understand some of the basics that they were sending me. These guys found a way to really start at the same place that everyone else did. Let's take whole venoms from an area, like Africa, and let's try to take whole venoms from a bunch of different elapids, so mambas and cobras primarily. Let's inject it into these camels. But instead of just taking off the plasma and using these nanobodies, let's pick out the best ones possible. And so they started off with tens of thousands of different nanobodies that they got from these animals. And now you could have 3,000 nanobodies coding for one single toxin, but they only wanted the best, right? They only wanted the most efficient, the best at binding, things like that. Yeah. So they did something called phage display campaigns. And this is where stuff gets crazy. I had to do it. This is where my knowledge kind of fell off a cliff, and I had to, find a way to somehow keep up in conversations with them where you are encoding the genetic code for each of those, 3,000 nanobodies is given to bacteriophages, or viruses that prey predominantly upon bacteria. They will present a physical copy of that externally, and then you can allow them to sort themselves out based on how well that nanobody that they're presenting actually binds to your target toxin. Because they carry the blueprint genetically for that antibody that they sorted out, the nanobody, you can take that genetic code and implant it into E. coli populations, And then just like, you have a batch of bacteria in brewing beer, you can have all these E. coli producing nanobodies in a cell culture in these huge bioreactors. And so your antivenom doesn't come out of an animal, it comes out of a spigot at the bottom of a bioreactor, which is just incredible. And it they took it even further and they said, okay, if I have a snake that has these 3 toxins and I've got these 3 nanobodies, if I stick them together and put them in the animal, does it save the animal's life? And it did amazing. I mean, wow. every test we ran was incredible. And we did a lot of the animal testing in Colorado, and it was just, it felt like every day we were doing it was just like a breath of fresh air just to watch how well it was working on some of these mice. It was incredible.
Right. I'm curious, just as a detail of that study, and you can continue on beyond there, but when you're doing these protection studies, are you dosing the venom via injection and then dose? I mean, everything's kind of controlled and
yeah.
Or are you testing different routes of venom exposure or areas where you're injecting? Like how is that? Is there a standardized protocol for that?
There's a couple steps that we take. Typically, first step is, does the thing work if you stack the deck in its favor?
Right.
So in this case, pre-incubation studies are that is the first thing we did. We have this product, we have this drug. It looks like it works in a test tube. Does it work in an animal? And the first things first, let's take the actual nanobodies and let's take the venom. Let's allow them to mix and interact, and then let's introduce them to the animal intravenously. And if you introduce the venom alone to the animal intravenously, especially like a mamba, they die within about 30 seconds. Right. It's kind of insane because we would inject regularly 3 times the lethal dose. So for that mouse, he would get his own personal dose, and usually it's like a 20-gram mouse. You would give him micrograms of a certain venom, and that is enough to kill it in roughly 30 seconds through the intravenous route of administration. It's actually kind of alarming when you give the injection, and by the time you put the mouse down, it is totally paralyzed. It's a little eye-opening as people who work with venom snakes.
You don't want to mistake that mouse for your hand.
And so we just want to see, does it work first and foremost? And we had great results. Almost everything worked just across the board for that pre-incubation. There was a couple of species that we were actually testing that we didn't think we would get 100% inhibition, and we didn't. And so it was nice to see, like, okay, our line of thought is correct, right? The ones that we aren't confident about are the ones that are not showing confident results. That's good to see. At least we're like on the right track in terms of how we're thinking about this. Right. We then went on to those kind of rescue assays. And these are where you are giving a challenge dose. In this case, again, 3 times the lethal dose. You cannot give it intravenously because they die way too fast. Right. And so you're moving to a more realistic model where you're either giving it sub-Q, so in the scruff behind the neck, or you're giving it IP or interperitoneal, where you're actually going into the body cavity and injecting some dose there as well. Right. And what we saw is if you wait 5 minutes, which 5 minutes seems super, super short, but this is a mouse. Things move real quick in a mouse from a pathology perspective. So you would give that 3XLD50, and you would watch symptoms start to appear really, really fast. And you would hope that 5 minutes came when the mouse hasn't died. And then you inject that IV kind of rescue cocktail of nanobodies, or even just a single nanobody if it's a more simple venom. And we would watch very clearly that you would have very good outcomes for the most part. And, not every trial was 100%, all the mice survived. But what was super important is we actually did the same things with the currently used antivenom in that region. And we were outcompeting it on almost every mark. Oh, nice. And that was like kind of like, okay, we're on the right track for sure, type of thing.
That's cool. So, wow. Yeah, that's encouraging when you're besting the positive control.
It's great to see. Great to see. And I think even now, something that excites me about this project, because obviously this is the tip of the iceberg. We got to keep going with it, keep doing a ton of stuff, is yes, we were out competing, but like not by huge margins. What's different about the nanobodies is that these are individual components that, like you were saying, it'd be cool if we can mix and match. We can mix and match these. So if we're working on a Dendroaspis, a mamba that's got some unique toxins in it, and the cocktail doesn't represent that, we can alter the cocktail to better reflect that mamba venom, and you will see much improved results from what you got with just like the standard cocktail we're using for everything else.
Right.
So,
and as far as you mentioned the stability, what kind of increases on stability? Because I mean, that's a big concern is, people that by the time they get a bite, their antivenom is outdated and maybe rusted shut or something in the vial,
so,
that kind of thing.
There's two answers to this. The first answer is biological. Biological stability is increased for sure, but biological stability of current antibodies is also phenomenal. Antivenom from 1970 is probably great. Probably works perfectly.
Okay.
As long as it hasn't been left out in the sun for 50 years, it's probably doing awesome. And cool. The other answer is pharmaceutically. The FDA puts expiration dates on stuff and they want you to replenish. And those expiration dates come from, whatever, safety measures that they did with those initial kind of trials. And a lot of it is, so you keep buying it because it's not like there's enough bites to actually, like in the US, especially to have this huge rotating, freezer full of antivenom, and you're not depleting the shelves. It hits that date and you have to move it off. And so that's really where a lot of that. Yeah, I know. It's too
bad you can't have that as a stockpile just in case kind of thing, like where it's better than nothing. I don't know. That seems like a tragedy.
Yeah. Private facilities do for sure. I know Jim Harrison over at the Kentucky Reptile Zoo, he gets bit more than most people, but he's often, statistically, he's definitely put himself out there, doing more venom extractions than almost anyone in the world, especially on a daily basis, man. Yeah. The guy's a machine. Right. And it's honestly kind of a surprise he doesn't get bit more. It really is a testament to his experience. And, he has tons of antivenom in him and Kristen Wiley. Like they hold on to every vial and they know that it can save lives still. It's not something that, you should ever throw away. Dr. Leslie Boyer, who was at the University of Arizona for the longest time running the Viper Institute, which is like a toxicology program down there. She had a walk-in freezer that was full of antivenom, and none of it was like current. It was all expired, going all the way back to, I think, the 1950s. It was an incredible collection. And when you tested that product, you can still inhibit a lot of toxins with it.
Right. Yeah.
So the biological answer, the pharmaceutical answer. Yeah.
That's cool. What about a vaccine for snake bite? Is that a possibility?
There's currently vaccines for dogs. And their efficacy is great for, I think, about a month or a few months. The trouble is, the issue with like venomation in general is, yes, you can have an immune response to it and you can stop it from an immunological kind of action.
Right.
But it's kind of inverse of a disease, where diseases, you get introduced to a viral load and then the viruses typically replicate, or bacteria, they typically replicate and then grow in abundance in your body. So they start slow and kind of snowball. Venom is the opposite, right? You are kind of hit with an avalanche that kind of trickles away as your body starts dealing with that issue. And so because it's not that kind of snowballing, your body's immune system cannot really like recognize an antigen and then mount an immune response before pathology start kicking in, like we can do with like diseases like a cold, where by the time we're hitting symptoms, our bodies are starting to mount an immune response and develop those kind of adaptive immune system reactions. Yeah, venom is just a different animal entirely, where
it's too rapid for the adaptive response to take hold.
And that's typically why you see that efficacy of the vaccine, is about a month, right? Because you have that really, really good immune response that you've learned from that vaccine, and then your body starts like putting it in the back pocket. But if you get bit, 3 months after that, then it has to pull that memory out of the back pocket and then reproduce the immune response. Right. And by the time that's happened, you should be getting antivenom.
Yeah, you don't have weeks to wait for your antibodies to respond. You have hours. Yeah. Okay.
That makes sense. It's a great like example of like why antivenom works. Antivenom is just an artificial immune response that you're mounting rapidly in replace of your own. That's all it is.
Right. Yeah. You're just putting antibodies in that would normally bind if you had time to create them in your body. Yeah,
pretty much.
Okay. Yeah. Interesting. And I'm curious, like evolutionarily, some of these animals that are resistant to bites, what are the squirrels or whatever that, kind of have an arms race, or, how are they, has that been investigated?
Or even seemingly the ability for the same species to be resistant to its own bite or one of its congeners.
Right.
Yeah. So probably the answer that's going to cover most taxa is bait molecules in general. So like I was saying before, these toxins can be inhibited either by destroying them, or you can remove their targets. You can also like fake out their targets. And so let's say a toxin is targeting collagen, and you have the molecule that's called alpha-2 macroglobulin floating around in your blood. That has 2 little bait regions of collagen built into this molecule. And so if one of these toxins comes and targets that, instead of it causing damage, it's like a tripwire. It triggers that and that break in the end and the protein, and then the protein folds in on it and traps it. And so you have this like kind of bait and switch and the ability to be a decoy. So instead of them hitting the important stuff, they're hitting this kind of fake trap molecule instead. And we see that in a lot of different animals where they develop decoy molecules to help kind of mitigate any impacts from venom being introduced to the system.
Right. Oh, that's cool. What about some sort of, pill you could take to introduce that into the bloodstream rapidly and bait?
Yeah. So that is, I think, a really hot topic. And I really think in the next couple of years, we're going to be seeing a lot of crazy stuff because, I mean, a lot of it's published. You can see that there's some good candidates for that exact thing right now. Right. The nanobodies are cool. The nanobodies are really awesome. The idea of a modular antivenom is super cool. However, no matter how good your antivenom is, it leaves a gap, right, from bite to hospital. It's still an antivenom. It still has to be administered in a place where it can be done, where if you do have a reaction, someone's there to treat that reaction to the antivenom. So there's a black hole there that has existed since the dawn of time from when you get, that bite to when you get treatment for that bite. And that is going to be kind of what is being targeted by companies like Ophirix, and this idea of a small molecule inhibitor that is orally available. Ophirix is a company started by Dr. Matt Lewin, who was like an MD, and he's got tons of accolades. I can't remember all of his titles, but it was him working in a bunch of other countries. I believe he was in Africa for a while, and just kind of seeing the impact of snakebite on some of the populations down there. And the issue was not that antivenom didn't exist in the country, it's that it's so far away. And I think a lot of us have had that moment in the back of our head. We're thinking, like, doing field work in the middle of nowhere, like, okay, how far is it to that nearest hospital? Like, we got to think about that every once in a while. And usually it's a few hours, it hopefully not a couple days away. But that is something that these people, that's their reality is, their treatment is so far away that do you even go in the first place? And, I was talking about, God, I got all the way back to just venom in general. You have a lot of venom profiles. You've got a lot of different toxins that we see across snakes, but you see a lot of similarities as well. We typically have, I could probably name 10 major toxins that are going to be like the most important ones across the world. And that's 10 for almost every species of snake to kind of have some similarity within that pool of 10. And so if you have a molecule that you can take just as a pill that attacks one of those 10, families, then you've got something that can effectively target some of these really, really bad bites that is readily available in places that, have that long distance to more proper treatment. And so Ophirix is spearheading a drug called Veraspodem. Veraspodem is very, very cool. It targets what we call phospholipase A2s. These are really, really common toxins. We see them in just about everything has some level of PLA2s. I mean, we have PLA2s in our body. But in terms of snakes, they can cause tissue damage. Like in an Atrox, they will cause like necrosis, so actually cause the cells to die off. But if you see it in a Mojave rattlesnake, that Mojave rattlesnake has a PLA2 that doesn't cause tissue damage. It becomes a neurotoxin in the Mojave rattlesnake. So it's a dimeric neurotoxin. Tiger rattlesnakes have the same thing. We see it in coral snakes, and like half the coral snake diversity in the Americas has significant amounts of the phospholipase A2. We see it in cobras all the time. We see different types of it in, gaboons and puff adders, and we see it going all the way through Africa. We see it a ton in Trimerosaurus, like species of green tree vipers, and that whole clade in Southeast Asia is full of phospholipase A2s. And so that is a huge one to target. If you've got a drug that can do that, and you can show those results and actually talk to the FDA and get something approved, that will be revolutionary. That will be a first of its kind. But that alone only covers one out of those 10 kind of major important toxins. And so what we're seeing is a few different not toxins, but these drugs that are coming out of the woodwork. And they're not just like random drugs. These are drugs that a lot of times have been studied, kind of similar to the drug I used in my master's research, for some sort of pharmaceutical purpose. A lot of them are anti-cancer drugs that never made it through the FDA's process. And so the orphan drug association, I'm not sure the exact title, they can take these drugs and then use them for other things. If they didn't work super well at this, can they work for this? And so Verapamil was one of those drugs that didn't really quite work for cancer research. It showed promise, but not enough to really push it through.
Right.
But the properties can apply here. And so that paired with something that could target, tissue damaging enzymes that we see as well, or something that can target other types of neurotoxins. If you can have a combination pill that could be a blanket coverage that you could carry in your pocket, every field herper would. I think we would probably get a little too risky, honestly. I think people would start freehandling a lot more. But just the idea of having that is pretty exciting for a lot of reasons. A lot of reasons.
We're definitely due for an advancement as well. Something that's less expensive that you could carry in your backpack. That'd be fantastic.
I've talked about this to a few people, like, how nice would it be to go to like REI or any outdoor store and see a snake bite kit that actually works? Like, it's not just a suction cup and a razor blade. Like, that would be awesome. I'm sick and tired of telling people like, oh, that thing you bought on Amazon sucks. It's horrendous. Don't actually use it, throw it away.
Nice.
So I want to give people options. Like, I think it's sometimes it's annoying and frustrating and honestly a little depressing. And sometimes for me, a bit embarrassing, because I'm in like the field of research for this, when people are like, well, what can I do if I get bit out in the field? And I say nothing. I'm like, go to a hospital. Like, I want a better answer than that for the most part. So I'm
right there with you. I study a lot of neglected tropical diseases that there's no treatments for, even though we've identified several that could work. They just, nobody's going to take them to market because there's no money in it. It's kind of a kick in the pants a lot of times, but what do you do? You keep going. Yeah.
One of these days, I think we're all going to be really happy to actually have a good answer for people and to set some minds at ease, which would be nice. Yeah.
Well, speaking of herping, do you get out herping much, and if so, what's been your favorite find herp wise?
Oh man, I don't get out as much as I used to back when I lived in Arizona. I had this moment when I moved to Colorado, and like after I'd been here a few years, where I was like, man, I had way more snake species at my fingertips down there. And I feel like I didn't see as many as I should have.
Neglected them.
Right. Our lab is very interesting in, I think, one of the best ways, where we sit at a crossroads of so many different fields, and we do so much. We have so many very like grad students that come into the lab, and we get to do so many cool things. I spend so much of my time, especially nowadays, cooped up in the lab, messing with mice, doing all these like very kind of, I don't want to say boring, but definitely tedious, like experiments where I'm just, putting stuff in tubes and then watching for a color to change and doing whatever. Where I get so antsy at times, especially during the school year where I have to teach and do other stuff. I want to go out and do things. And, we're lucky enough to where we have field projects ongoing. And if I get a spare week, I'm like, cool, I'm dipping out to the western slopes and meeting up with our field crew and looking for con color and
Nice.
in some of those areas. So every once in a while I get to, kind of stretch my legs a little bit and go out into the field. Just like last month, I met up with our field crew out in Grand Junction area. They're doing a huge study right now on con color across the entire range. It's just a big, let's see how the populations are doing. Let's do mark recapture.
Cool.
So there's a ton of cool work. And it's always fun to meet up with people who are experts on this one species.
Yeah.
And just to be like, all right, point me in their direction, right? Take me to where they are. I want to find stuff and take photos.
Yeah.
And so it's very low stress herping sometimes.
Yeah.
And then of course, just going to talk to them about their, all the stuff that they're finding out there is so much fun.
Yeah.
I get to do my cruising out in the plains every once in a while when the weather is good and wants to work with us.
Right.
But yeah, I miss being in Arizona where, herping was, I don't want to say easy, but definitely more accessible. I think you've got more options down there, especially year round. And then, being over in Colorado is kind of, it feels a little boxed in sometimes for the herps. But yeah, I get to go out whenever I get the opportunity to. I've done a few, trips where I've gotten lucky enough to be with some people where we've done Taiwan a bit. We spent a couple weeks out there, and Taiwan is, man, it was my first time in Asia. And after that trip, I just couldn't wait to go back. I mean, it's an incredible place, incredible country. I think we got to see so many cool parts. We did incredible out there. We got Dinokistrodon, Anabophis, Makazaya in one night, which is incredible. I mean, finding I think one of the holy grails for us out there was the Ovophis species. I mean, it's just incredible seeing them out there, meeting up with locals.
What's the common name on those?
I believe it's Alishan habu, otherwise known as the strawberry snake.
Okay.
Latin name is Ovophis Makazaya, which is one of the cooler Latin names.
I don't know my Asian viperids very well.
I really didn't know that much about it. I think one of my big targets was most definitely Naya-Atra, the Chinese cobra, and on Taiwan, just because seeing cobras in American is always cool. Always cool. Yeah, sure. And then Dinokistrodon acutus, the Chinese cottonmouth. Was just awesome to find as well. But yeah, the strawberry snake, at least that's the common name that we were calling it at the time, was something that I really didn't know much about. It's a very kind of not reclusive species, but one that's really strange. You would only find it, the locals were telling us, you only find it when it's raining out. And that's when it crosses the road, which is, obviously a little different. I'm used to going out after a rain. So kind of switching up, our styles a little bit, but yeah, it resulted in some really awesome finds. It's a pretty incredible trip. With that same group, I've done Peru on the western coast of Peru, which is the part of Peru don't herpers don't go to the western part very often. I think a lot of times you think about Peru, you're going into the Amazon. And we did not do super well. I think most of us just got sick, which I mean, you have to have one bad trip at least once in your life. And that one was pretty rough. But then, hopping over the next time into the Amazon and, getting to help out a researcher from Florida State University down there in kind of that Napo River Valley was really awesome. Stumbling across a huge lachesis on the trail was just nice. I think a holy grail for a lot of herpers within the Americas. That's just that's something that is just incredible. That's a night that I'll just never forget. But yeah, I'm looking forward to doing more. I've got a trip coming up in August. I'll be going with the Asclepius Snakebite Foundation to West Africa in Guinea. Oh, cool. And this is a team where it's mostly actually MDs and medical professionals who go to the snakebite clinic out there, and they assist the Ghanaian the Ghanaian physicians in treating snake bites. And, they work to kind of help mitigate, snake bite mortality rates out there. But we also get to work with them and collect venom from anything we find. So the locals will actually call us for snake calls. Obviously, most of the times it's puff adders. I mean, that's the predominant thing. It's across almost all of Africa. Green mambas, the Western green mambas are also very common out there as well. Cool. And the clinic has actually been brought in the past couple of years 2 black mambas, which for Guinea is incredibly rare.
Yeah.
And I believe one of them was the first live black mamba caught in the country. And so like the fact that facility has enough really good connections with the locals to work with them on that level is awesome. So I'm excited to just go out there. You know, it's tough herping in those regions. But I mean, just getting to be there in that environment would be awesome. And to like watch these physicians really, really work on the ground and talk to them and, you know, have that discussion of like, hey, we're developing these treatments. What types of data do you as a physician want to have? And, you know, like what do you want to see in your treatments as a physician? And so that's like the big goal of the trip. And then also, you know, mambas is awesome. Like that's just hard to ignore.
Some of the bittis as well, you know, gaboon viper or something would be really a cool thing to see in the wild. I know one of the MDs is actually a huge herper. His name is Jason Folt. He is a very, very adamant herper. And sometimes like when he's down there, he's like, oh, well, there's an Atheros spot that's like 5 hours away. We could make a day trip. And the other MDs who are like not snake people are like, I'm not doing that. Like, what's wrong with you? Right.
And so I know
waiting for the herpetologist to get there. So
him and I were talking about it a little bit. And he's like, yeah, there's a local guy who told me that he's got this one spot. And I was like,
oh, cool.
I'd be down. To
go. So, well, and use those connections while you know you can, and have those. Us lay folk don't get those kind of connections very often.
I know it's a crazy place. This is a connection that's very surreal to me. I was mentioning I've been wanting to do venom for a long time. Yeah. And I've been doing anti-venom stuff almost just as long. Back when I was working at the sanctuary, we actually had a cobra bite back in 2017, 2016. And it was like one of the first cobra bites that an Arizona physician had ever treated. They did an amazing job. Problem was they had to fly in anti-venom from Denver. And, you know, as a facility that has a bunch of venomous snakes, that's not great. And so then we started up the Phoenix Herpetological Sanctuary Antivenom Bank, which is one of the largest antivenom banks in the American Southwest. And being a part of that project was so cool. I got to meet so many physicians and really learn the ins and outs of like, okay, what is antivenom? Why is it doing all this stuff? Right. And we got to a point where I met up with Dr. Leslie Boyer, and her and I talked a lot just about the whole world of snakebite and what are like the challenges facing it. And she was like, there's a clinic in Guinea that is really cool. And so now fast forward 10 years, Leslie Boyer is sitting on is like one of the medical advisors for Ophirix as a company, working on Veraspita. But I get to work with her on that project and also like do this clinic. And it feels very full circle, very surreal at times. Yeah. And man, part of me wishes that a lot of these MDs would go to more of these like herp conferences, because I like having conversations with them is super eye-opening, especially as like people who, we're out in the field a lot and just getting to do that. But man, hearing their perspectives on things is so cool. And I've tried to convince them to come to like biology of the pit vipers and biology of the snakes and a lot of those conferences. And they're like, oh, well, like maybe next time. I'm like, you're missing out. You're missing out. So,
wow, man, what a great field you're in, and how it's some really exciting work. We really appreciate you coming on here and talking about this stuff. It's so fascinating and just exciting, just thoughts of what could be.
Yeah. And I think, like I mentioned, we're definitely, and you said it as well, we're in a turning point, right? This is like definitely the corner that we're kind of coming around right now. And man, give it a decade. And I think this field is going to look a lot different than what we currently have, for the better. Thank gosh.
Yeah. It's about time, right?
Man, 100 years in the making at this point.
A lot of time with horse serum. Yep, exactly. That's exciting. Well, good luck to you, and hopefully we see those advancements sooner than later, right?
Yeah, it's the goal.
Cool. Right. Rob, did you have any other questions?
No, this has been fabulous.
Yeah.
Well,
where can people find or track your progress or see your, aside from Google Scholar, see your publications? You have any place you post or?
I mostly, in terms of social media, just on Instagram. Should be pretty easy to find for the most part. Toxic_Sam will get you there.
Okay.
I get a lot of weird looks when I mention that name sometimes. But yeah, I use social media sometimes. I think, like many herpers, my social media is almost entirely snake pictures and skeletons that I articulate on occasion. That's about it.
Oh, very cool. Yeah.
So, yeah.
Yeah, I'm I've got a little bit of a dead zoo behind me. I don't know if you can see it, but it's on the shelf there.
That's awesome.
By the way, but yeah, like some different skulls and things. But yeah, I thought that would be really cool to do an articulation or some like wood burning or carving of a skull would look really neat. Too.
Yeah.
I got a really cool Aboriginal painted crocodile skull that I brought back from Australia with me, which is pretty sweet. Yeah.
Yeah, that's something I need more of. I think, being a grad student, you learn to move around light in terms of belongings. I'm excited
a heartbreaking story of the grad school student moving. We had a grad student at Utah State that he had a collection of things that he'd found out in the wild or whatever, and had them as pets in his apartment, and then he got a job somewhere, finished his PhD, and then just euthanized all his pets, put them in the collection at Utah State, and left. I'm like, oh my gosh. That's so. That was one of the things that made me decide not to be a herpetologist. I'm like, I can't be that callous. I'm sure not all herpetologists are that way, but the pickling and that kind of got me a little. I'm like, I don't know if I could do that too well.
Yeah. I mean, every once in a while, like maintaining a beetle colony kind of gets on me. It's a little annoying,
right?
But when they take a skinned animal down to a skeleton in 24 hours, you're like, all right, I kind of love you guys. This is
very
phenomenal.
Helpful. Yeah. Oh, very cool. Well, thanks again for coming on, and
it's been a pleasure.
Yeah. I'm trying to think if there's anything really cool, at the end of the show, we like to kind of say if there's anything really cool that you've seen in herpetology, herpetoculture, anything like that in the last little bit. I'm excited about they're going to air a program on the Kimberley on the 17th of June. So I'm really excited to see that. I hear there's footage of rough scale pythons and some other Kimberley endemics. So on PBS in June 17th. So I'm looking forward to that next week.
That's awesome.
Yeah. So it should be fun.
Yeah. I don't know. I think, there's a lot of advancements coming up and as follow-ups to some of our research that I'm hoping will get the light of day here soon. So in terms of media, that's where a lot of my attention has been. So, media in terms of, scientific publications. Yeah.
We'll keep an eye on your profile. See if you got any publications coming out soon. Good stuff. Yeah.
But I pre-ordered someone's field guide to the reptiles and amphibians of Utah.
Oh, very cool. Yeah.
Heck yeah.
Yeah, I'm glad to see it's finally out. And I think they even put a price to it. It was like, was it $21 or something?
Yeah, $20.95, something like that.
Yeah.
Wow.
Bob kept it low price. That's impressive. Yeah. I got 2 advanced copies that I've had a chance to peruse. It costs $50 to send one to Nipper, so I'm not sure how we want to work that. I might just wait till he's in country and give him a box to take home or something. But yeah, it's a little tricky there. Sorry, Nipper. But yeah, excited to have that out and hopefully in hand sooner than later. We're thinking mid-July is when the main shipment arrives from China, and then I'll pick up a bunch of boxes at the IHS meeting. Do you happen to be going to that, Sam?
Unfortunately not.
Oh, okay.
I'm going to, I had time for one conference this summer and I'm doing JMIH instead.
Right.
So got a lot of friends going down to IHS, but unfortunately couldn't make it.
Very excited to be there. I mean, it's the 50th the 50th one, so it should be a pretty big production, but looking forward to it.
And not to mention the new library down there is just
it's fantastic.
Gosh. I was down there for biology of the pit vipers last year. which it was super cool to be down there. But during a conference, it's a bit noisy in the library.
Right.
And I kept saying to myself, like, I got to come back when it's not a conference.
Jeez. Yeah. We've been there a few times where it's just been, 5 or 6 of us at most. And just jaws down on the ground. And Bob pulled out some books that were, worth a lot of, I think he, paid tens of thousands of dollars for some of these books. And it's pretty cool to see these old, prints and lithographs and stuff in the books. It's amazing.
Yeah.
What a collection.
His collection never ceases to amaze me what he's got down there.
For sure. Good stuff. All right. Well, thanks again. And we'll thank Eric and Owen and the NPR crew for hosting us. And we'll catch you next time for Reptile Fight Club.